Hello children, I hope you're staying safe at home. To continue with economic importance of bacteria and fungi, today I'm going to talk about the useful role of bacteria in medicine. Now, bacteria are used in medicine to produce antibiotics, serums and vaccines, okay? Now, when we talk about an antibiotic, we are talking about a chemical substance, okay, which is produced by a living organism. And what does it do? It stops the growth of disease causing bacteria and fungi, okay? There are different types of antibiotics today which is used in the treatment of different types of infections, okay? Certain antibiotics are streptomycin, chloromycetin, tetracycline, penicillin, to name a few. Okay, there are others also, many different types of antibiotics. Okay, now the first produced antibiotic was penicillin. Okay, in 1929, Alexander Fleming saw that some bacteria, like he was working with some bacteria and he had grown some bacteria in a culture in his lab, and suddenly he saw some fungus growing on it, a mold which was known as Penicillium chrysogenum. Previously it was also known as Penicillium notatum. Then he studied it and he saw that this mold, that is Penicillium chrysogenum, produced a substance which did not allow the growth of many bacteria. And then the substance was named penicillin. That was the first antibiotic that was used in medicine. And remember, penicillin came from a mold, a fungus, and it is not from bacteria. Okay? Then after that, streptomycin was discovered by Selman Waxman in a similar way, and it came from bacteria. Okay, this one came from a fungus and this came from bacteria, bacterium, Streptomyces gracius. Okay, and now I have underlined these. Why? Because they are all scientific names. I hope you remember how you're supposed to write the scientific names. Okay, there are some well-known antibiotics, okay, and they come from different sources and they treat different diseases. Like you have penicillin, it comes from a mold, Penicillium chrysogenum. It treats tetanus and diphtheria. Check your text. There is a table on page number 86. Okay, and please go through. That table is very, very important. You have to learn by heart. Now, next, moving on to the criteria for a good antibiotic. It should be able to kill different types of bacteria, not just one single bacteria. Okay, if you see an antibiotic medicine, you will see the word broad spectrum written on it. Okay, broad spectrum means it can kill many bacteria, not only one bacteria. It should not produce undesirable side effects. There should be no side effects. Okay, and of course it should not kill any useful bacteria of the organism of the host. Okay, now there are other uses of antibiotics as well. First is it is used as a food preservative, especially for fresh meat and fish. You know that food, especially fresh meat and fish, is spoilt by bacteria. So if antibiotic is there, the bacteria will not grow, and so fresh meat and fish will be preserved. Okay, it is also used in treating animal feed, like in the case of poultry, chickens. In their feed, antibiotic is put in so that these chickens do not get infected by any bacteria, and it is also used for controlling plant pathogens. Now, plant pathogens are bacteria which causes diseases in plants. Okay, now moving on to serums. When we talk about serum, we are talking about blood plasma. If you remember, blood plasma is the liquid part of blood. It is the liquid part of blood from which fibrinogen has been removed. Now, fibrinogen is a substance which is present in blood which causes blood to clot. Okay, now if this is removed, especially when you have a cut and you start to bleed, you notice that the blood clots. It is due to the presence of fibrinogen. Okay, so if we remove the fibrinogen, that means the blood will not clot. So what will remain behind? Only the liquid part of blood will remain behind. But the liquid part of blood will contain different types of chemical substances like antibodies, antitoxins. Okay. Now, what are toxins? Toxins are poisonous proteins, and it is released by pathogenic bacteria. Pathogenic means disease causing. Okay, disease causing bacteria are known as pathogenic bacteria. Okay, and so toxins are poisonous proteins which is released by pathogenic bacteria when they are growing during their growth or upon their breakdown after they die. When they die, they break down and they release toxins, that is the poisonous proteins. Okay, and then moving on to antitoxins. Now, antitoxin, anti means against, so antitoxin means acting against the toxin. Okay, so these are substances which is produced where? In the bodies of animals, which reacts with the toxin, that is, it acts against the toxin produced by the germs, disease causing germs. Now, moving on to serum. Now, remember, serum, that is the liquid part of blood, contains antitoxins of a particular pathogen, one bacteria. Okay, and serums, where are they used? They are used as a prevention. Okay, as a prevention against bacterial invasion. Now, to prepare a serum, a small dose of bacterial toxin is taken. Okay, bacterial toxin is taken and it is introduced into the body of a healthy animal. Okay, so now what happens in the body of the healthy animal? Like horse or cow. Okay, so the body produces antitoxins. That is, it will start producing antitoxins. And many times this is injected into the body of the healthy animal, and after some time the blood is taken out. Okay, and on chilling, that is when you cool it, the serum separates out from the clot. The clot is separate and the serum is separate. Supposing you take it in a test tube, okay, and then you put the blood of the animal here. After some time, you will see the blood clot and the liquid. So here you have the blood clot and then the liquid is the serum. Okay, so snake bite treatment also involves injecting antivenin, which is produced in the blood of horses. Okay, that is the horse, into the blood of horse, the poison of snake is introduced, and then the horse is a very strong animal, so it produces antivenin against the venom of snake. Okay, so antivenin is produced. So that is exactly what is introduced into a person who is bitten by a snake in the hospital. Okay, now genetic engineering has also made it possible to introduce human genes in certain bacteria like Escherichia coli, E. coli. Okay, so these bacterial cultures grow very rapidly. So the particular gene dependent products are extracted from these bacteria. So the hormone insulin was the first such substance which was produced by E. coli. And there are certain serums, compounds produced by genetically modified bacteria, like you have the blood clotting factor 8. Okay, this is used in the treatment of hemophilia A. Okay, and factor 9, we're talking about the serum. Okay, this is used in the treatment of hemophilia B. Now moving on to vaccine. Okay, all of you have been vaccinated when you were small. Okay, now when we talk about a vaccine, we are talking about a preparation which consists of weakened or dead germs. Actually, when you are vaccinated, what is put inside your body? Weakened or dead germs, so that your body can produce antibodies against these. And later, if you get this disease, your body will be ready to fight against the disease. So when we talk about vaccination, we are talking about introduction of weakened or dead germs into the body for developing resistance or immunity against the future attack of the disease. Okay, now vaccines are available for different types of diseases such as cholera, typhoid, measles, chickenpox, and so on. Okay, all of you have been vaccinated when you were small. Okay, and so what happens when you get vaccinated? What is introduced into your body? Germs. Either they are weak or either they are dead. And what happens? You get the disease in a very mild form. Okay, and then your body will produce antitoxins, and the antitoxin will remain in your body, and it will provide immunity against any future attack of that particular germ. Okay, now there are vaccines obtained by growing bacteria. First is where you use killed bacteria. It is TAB vaccine, which is used for typhoid. Okay, and living but weakened, very weak bacteria. Okay, example is BCG, Bacillus of Calmette and urine, which you'll be learning later on. Okay, BCG vaccine, it is used for tuberculosis. Okay, now next we come to toxoids. Now what are toxoids? These are inactivated toxins. Okay, it has not yet been activated of a particular bacteria, and it can still stimulate or it can still produce antibodies. Okay, it can still produce antibodies. Even though the toxins are inactivated, okay, it is taken from a particular bacteria and it can still produce antibodies, such as for producing immunity against now diphtheria, and tetanus. That is when you go to get a tetanus shot or a TT shot, as you say, what are you getting injected with? You're getting injected with inactivated toxins, which will produce antibodies against diphtheria and tetanus, okay, whichever shot you take. So we will stop here today. Stay safe, and if you have any doubts, please WhatsApp me. God bless you.